Good evening, esteemed guest, our valued audience, and my dear colleagues. My name is Yasha Niriti, and I am currently holding a position as a scientist at Biocon Limited in Bengaluru, India. On behalf of Neighborman Bioinformatics, I'm thrilled to welcome all of you for today's webinar, where we will explore the exciting prospects of QSAR and delve into job-demanding fields of drug discovery. So please allow me first to provide a brief introduction to Neighborman Bioinformatics. Established in 2019, we are a globally recognized bioinformatics service company that specializes in offering training, project outsourcing, and consulting services in the field of bioinformatics. Furthermore, we offer expert assistance in various areas such as structural bioinformatics, cheminformatics, computational drug discovery, molecular dynamic simulation, omics, and related domains. For additional details, please visit our website, www.neighborman.com. I'd like to, before moving ahead, I'd like to introduce to our webinar series, where you can delve into the intersection of bioinformatics and biotechnology through our bi-monthly webinar series. Our webinars are thoughtfully crafted to shed light on most recent trends, groundbreaking discoveries, methodologies, and the dynamic relationship between bioinformatic application in the realm of biotechnology. Likewise, we explore how advancements in biotechnology can impact and shape the field of bioinformatics. So are you enthusiastic about staying informed or perhaps even participating in our webinar series? If yes, then stay updated and discover more, considering and subscribing to our website, www.neighborman.com/webinars. So now, before beginning towards our webinar, there are a few important instructions that is to be followed. Please listen to it carefully. So we begin with the first one, where the chat will be accessible to participants only during the Q&A session. To ensure that only registered participants receive the certificate, please follow the steps below. So send us an individual direct message on our Instagram page to request the feedback link for the certification. And during the webinar, we'll address requests for feedback link between 6 to 7 p.m. IST. And the same link gets activated from 7 p.m. IST. And we will be accommodating Instagram message requests up till 11:50 p.m. IST on Sunday, 3rd of September, 2023, that is today itself. All right then. Now we will begin with our webinar. So I'd like to introduce our guest speaker today. We have Dr. Chandrasekhar Ponnusamy as our guest today. Dr. Chandrasekhar not only holds the role of co-founder at Neighborman Bioinformatics, but also stands a distinguished scholar and educator in his own right. His remarkable contributions to the field of bioinformatics have been shaped during his tenure as a teaching and research associate in Vellore Institute of Technology. Additionally, during his postdoctoral experience at IIT Madras, Dr. Chandrasekhar specialized in QSAR, quantum mechanical, and molecular mechanical studies. His focus included the prediction of anti-cancer activities derived from nanoemulsion based on spices. With a genuine passion for academia, Dr. Chandrasekhar has authored 10 peer-reviewed journal publications and contributed a chapter to a scholarly book. His commitment to sharing knowledge is further underscored by his involvement in freelance tutoring endeavors, where he imparts his expertise to students preparing for competitive exams like GATE and NEET in India. So with great respect and enthusiasm, I'd request Dr. Chandrasekhar to take it over. Sir, the floor is yours now.
Thank you, Yasha, for a very great introduction. And thank you, Neighborman, for the opportunity. So hope you guys will enjoy the forthcoming session. So as said by Asha, so today we are going to see the webinar on unlocking opportunities with QSAR, the job demanding technique of the drug discovery. Fine. So it would be fine. I'll stop the video so that it will be visible for your better understanding. Fine. The sneak peek of today is Introduction to Computer-Aided Drug Discovery and the importance of QSAR in CADD. And we are going to see the major types of QSAR models and the career opportunities with QSAR and related fields. So guys, you might have aware of what is CADD, like computer-aided drug discovery, and the importance of how QSAR is going to a magic bullet, acting as a magic bullet in CADD. And we are going to see the major types of QSAR models, like particularly regression models and classification models. And of course, we are going to see the career opportunities at the end of the slides, which is the cream of this presentation and the related fields. Drugs. Coming to drugs, medicinal drugs are the one which is used for a very long time. So man is fond of drugs for a very long time, right? Like right from his caveman time till now, man is using drugs for several purposes. For example, the purpose such as curing, right, halting the disease or preventing the disease, or to cure the ease of the symptoms, correct? He's using drugs for several purposes. Right from the time of caveman to computer man, today we can design drugs and we can manipulate the drugs and perform a number of analysis. So everything is in the way of digital now, right? Fine. When you go for a drug in a market, so guys, I think this session will be interactive. It'll be so helpful for us. So have you guys, you might have an experience of buying medicines in the market, right? What is the maximum cost you have bought drugs? Anyone over there? What is the highest price you have purchased the drug? A tablet.
From $1 to like $10.
Yeah, to $1 to $10. Fine. Like 80 or 100 in the case of INR, Indian national rupees. But do you know a drug may cost even lakhs of rupees? That is cancer drugs. For a cancer drugs, like per cycle, we need at least eight cycles, right? Per cycle, it costs 1 lakh rupees. And it costs even more, like 17 lakh rupees. So definitely these drugs are cancer drugs. So the question comes here, why drugs are so costly? Because drugs are costly because the drug discovery is a costly process. It's a time consuming and a tedious process. Nearly for an average, a drug candidate from laboratory to pharmacy takes nearly 14 years and $1 billion. And not all the drugs are ending up in a high success rate. Most of the drugs end up in low success rate. Fine. End up in low success rate. The reason is, see, most of the time in drug discovery lied over the preclinical trials. See, a lot of the drugs have been moved to trash the preclinical trials itself. That's the reason. As Lord Ram went to Manavash of 14 years, nearly a drug to come into a market requires 14 years. Yeah, as I said before, it is two things. One is time consuming, another is tedious. The reason for time consuming is in drug discovery, we are involved in checking a drug, like any molecule from a plant or an animal or any chemical compound, chemical substance have to pass through two things, like in vivo and in vitro. Okay, like it takes 5 to 6 years. And clinical trials, again, 3 phases of clinical trials. It takes months, years, and several years, like 1 to 4 years. And even after passing through phase 3, it has to go through drug review, FDA approval, and post-market monitoring. So a number of processes have to be carried forward. And apart from that, those drug process discovery is also tedious, as I said. For example, let us start with 10,000 compounds in phase 1, like drug discovery process. It will end up in 250 compounds in preclinical development. And moving on, in stage 3, it will be like 5 compounds, right? 10,000 to 5 compounds. And as I said, there are 2 important terms you have to know in drug discovery, that is pharmacodynamics and pharmacokinetics. So what I said here is pharmacodynamics and pharmacokinetics. So it's quite simple. Okay. See, if I consume a drug, I have to know what the body does to the drug. So when the drug enters the body, our body will react as like other external things which is entering the body, our body will react, right? So what body does to the drug is pharmacokinetics. So here only we will study about the 5 important properties called admit. So anyone out of the audience can say what is admit? It's an abbreviation. An abbreviation for what? Guys, anyone over there?
Yeah, can you say?
Yeah,
Okay.
please.
So it's absorption,
Correct.
distribution, metabolism, excretion, and transportation.
The
Is it
fourth is
okay?
right. The last one is, can you repeat again the last one?
I think transportation.
No. More than transportation, the very important property is toxicity.
Toxicity. Oh, yeah. Sorry. Yeah.
Yes.
Thank you.
Thanks for your participation. So as ma'am have said, it's correct. Like it starts with absorption, distribution, metabolism, and excretion and toxicity. So any drug in the market have to be checked for all these properties. So these 5 properties are none other than, as I said before, they are PKK, like pharmacokinetics. But what is PKD? That's the actual role have to be happen, like mechanism of action of any drug. So it's an ulta of it, what I've written here. That is what drug does to the body. What drug does to the body. So once the drug reaches the active site, we should know, right, how it's going to act over. Okay. So by learning all this, only a drug will reach to the regulatory approval stage. Drug does to the body. Fine. Hope you guys clear on this. So with this understanding, let us move forward. Okay. Here comes the actual magic bullet. So computer-aided drug discovery is a magic bullet, as I said, because to test, we need a lot of compounds to find a drug, right? 10 to 15 years and 500 to 100 million US dollars are invested. But how CADD is going to help us? It helps in the initial stage, like virtual screening, and it will help to select the leads and lead optimization. So these are the actual roles. By doing this, we can save nearly 2 to 5 years and reduce this amount, like 100 to 500 million US dollars can be saved. So instead of, it's quite simple, right? Instead of using 500 compounds out of 1 lakh compounds, I can directly give to a clinical researcher of only 20 compounds. Fine. Of my organism of interest, from where I'm going to derive the drug. So to highlight this, it reduces the cast time, it reduces the animal use in the experiment, improves the success rate, and improves the understanding of drug protein interactions, what influence it is going to make with the drug. And in the case of structural bioinformatics, the 3 mantra is there, like 3 is an hierarchy is there to know better of a function more than sequence. So whatever the thing, it starts from sequence, structure, then it goes to function. So knowing a structure will help us to find the function of any molecule in that easy. That's the reason even AlphaFold, you might have heard of AlphaFold, right? The AI technology of Google, it's highly focused upon solving the structures, like modeling the structures. So computer, again, in the case of CADD is a huge field. Fine, the CADD process, in which the 3 major methods are there, like ligand-based, structure-based, and system-based. Before that, let me, this is the actual thing what we have seen just before, a couple of slides back, like target discovery, that is identifying a disease modulating target protein, and hit identification, lead optimization. And as I said just before, like pharmacokinetic properties and evaluating their dosage and efficacy in the clinical trials. Okay. Again, what is ligand-based? What is structure-based? What is system-based? So you guys, if you heard the term called bioinformatics, you might have heard this fancy terms called molecular modeling, right? molecular modeling, protein structure prediction, docking. I hope that out of the participants, many of them might have involved in this docking things and all. Okay, how many of you have an experience of QSAR before? Like in the case of ligand base, you just can give a thumbs up in the group so that I have an idea of explaining much or less. Okay, I got one thumbs up over the group. Fine. Okay, at least I assume that many of you have interest over learning this. So it's quite simple. What you have to understand here is what is known to you. From there, we can start our drug discovery process. For example, my lab is involved in knowing the compounds of a particular plant. So we are the group interested in knowing the compounds of curcumin, like from turmeric. I want to know all the compounds, and my lab is working over there. Then I can go for ligand based, because I can do a simple GCMS or LCMS and know the compounds, and I want to know what is the actual target over it. So in this case, to know the activity, I can use QSAR modeling or computational chemistry or chemoinformatics profiling. Or in Ulta, I'm from the lab who working on collagen protein and leather industries from leather background. I know the structure. Then in this case, I have to look for the suitable drug molecule which influence my target compound. And there, due to the evolution of omics, another thing called network pharmacology, proteometric modeling, and pathway analysis is a booming field now. So our QSAR modeling lies under a category of ligand based. Okay, this should be answered by everyone, I guess. Which is the mother of science? So we have many scientific domains, right? Which is the mother of science here? Any guess? Chemistry? Is it physics?
Mathematics.
Yeah, mathematics. Maths is the mother of, correct. Maths is the mother of science. So every solution to the problem lies in maths. So that's what QSAR also adapts itself. Like we are going to convert this structure to numbers, right? As the name suggests, quantitative structure activity relationship. Okay, the structures cannot be understood by the by computer. See, again, the very word computer means count, right? We might be aware of this. The very word computer is to count. So computer knows only by what? As digits, as binary digits. To make it effective, to make a full use of it, we have a concept called descriptors. So descriptors are none other than, and they are the mathematical notations of a particular structures, of particular compounds. Okay, so QSAR is a technique where we use computational modeling method for revealing the relationship between a particular structure property of a compound and its biological activity. Fine. So descriptors are treated as independent variable, like these are the descriptors. So it's a function of physicochemical properties. For example, I want to know whether my compound, for example, curcumin, have an anti-cancer activity or not. I can check with its physicochemical properties. Okay. So based upon the physicochemical property, I can predict its activity. Right. So descriptors plays a huge role over there. To make it simple, using QSAR model, that is a mathematical model, trained with known activity, I can predict the unknown activity. So I'll give a notation over here, an analogy over here. See, imagine is a 2-year-old baby. Okay, it never have seen a dog or a cat before. So what parents are going to do, and the baby's parents are keep on training the baby, like showing a dog, okay, of different kinds of dogs, and informing it it's a dog, it's a dog. The same way it is the parents are training the baby with cat. So by this learning, the baby can easily recognize a cat or a dog easily, right? The same thing happens here. We are going to train the model with the structures of different structures. By training the model with the different structures of a particular activity, for example, a dog will bark, a dog will have a long lengthy tail compared to cat. Like that, I'm involving with a lot of features of the dog, right? The same way, I'm just suggesting a particular function like anti-cancer function is based upon this kind of a chemical compound, this kind of a chemical moiety. So it can be easy to find the prediction of its activity. Fine. To know it very well, we have to have the knowledge of molecular dimensions. Again, so what dimensions we guys are living? This is so easy, right? When we go to a movie, you guys have an experience of watching Avatar movie in 3D, right? Like 3D. What's called 3D here? It's quite simple, right? X, Y, Z. Okay. Like length, width, and height are 3 dimensions, whereas area is just 2 dimension. And 1 dimension is just a length, whereas 0 dimension is just a dot. And we have a 4 dimension that like tesseract. Even though the 3D is there, like X, Y, Z coordinates are there, and it is keep on moving, it's called 4 dimension. Right. So this knowledge is important for understanding descriptors. So as I said, a structure can be used to train a model, and the model can be used to find the function. So what kind of training we are going to give for the molecule is based on the descriptors. So in QSAR, the understanding of descriptors is very, very important. Like 1D descriptor, like molecular weight, or what elemental composition are there, like how many carbons are there, how many hydrogens are there. These are 1D descriptor. The same way 2D descriptors, mostly used descriptors like topological descriptors or hydrogen one acceptors, donors, solubility. These are all 2D descriptors, and 3D descriptors like polar surface area, molecular surface area, volume. Yes, 3D descriptors. And we have, based upon the different research groups, certain descriptors are there, like Abraham descriptors, moles of descriptors, so and so. So these descriptors are none other than they are mathematical representation of the molecules, as we discussed a few slides back. And again, the trending field now is machine learning, right? So the machine learning in drug discovery is saying about creating machine learning models, okay, of several drug types and databases, as said in Paulo et al. 2021. So they have clearly said about how the molecular descriptors and mathematical models, molecular descriptors will help to create a mathematical machine learning model. And from that, we have predicted Paka performance and conclusions. So coming to QSAR regression classification models, three things are trending now, like supervised, unsupervised, and reinforcement. Supervised, unsupervised, and reinforcement. For example, supervised learning means, as I said, parents' baby are training their the parents training their baby with the known values, right? The parents know already how a dog will look like and how a cat will look like, and they are informing their baby in that way. But imagine the child is discovering the cat and dog by itself. So that is called unsupervised. So here a supervisor exists, here the unsupervisor. No supervisor exists. Okay, under the supervised category, that's out of our interest today, like regression, decision tree, random forest, classifications. These are some of the famous names, like KNN or classification algorithms. So guys, are you aware of the terms called regression and classification? Can anyone out of the group can say what is regression, what is classification? At least you can answer the question called what is classification, right? Don't feel me odd, make me odd or alone. Is that you guys are interacting, listening to which you guys can understand what I'm saying?
Regressions are continuous values.
Okay.
Classifications are 0 or 1, mainly like yes or no type.
Correct. So classification is yes or no type, right? Thanks for the answer. So again, so may I say in this way, which among them is qualitative and which among them is quantitative? So definitely classification should be qualitative, yes or no type. Okay, as one of our participants have said. To make you simple, I am asking the question, it's hot or cold, the temperature. What might be your answer? Either hot or cold, right? So this is classification type. Okay, again, as a supervised learning algorithm, it is categorical, yes or no type. Whereas regression is a kind of continuous variable. For example, if I ask what's temperature, is hot or cold, I'm saying it's 24 degrees Celsius hot, right? 30 degrees Celsius hot or 84 degree Fahrenheit hot. So these are regression. I say in biological terms, if I'm asking a particular drug compound is active against cancer, if I'm saying an answer called active or inactive, it is called classification algorithm. It means classification type. I am saying it's IC50 values. It is a particular micromolar active, 5 micromolar active or 3 micromolar active. If I'm saying that statement, it is regression. Okay. And again, to assess the model, we have several methods like root mean square error in the case of regression and percentage of correct classifications like true positive, true negative, those classifications in the case of classification, right? Again, model or feature selection and validation is a huge process. If I have an anti-cancer thing, which is a concentration for 50% of the maximal inhibition of experimental cancer proliferation, so that can be assessed. That is, the biological activity can be assessed, as I said a few minutes back. And in the case of linear regression, we have to design the model and validate the model. Each and every descriptors have to be validated statistically. By design, designing the descriptor determines the quality of your model. Okay. Then again, we can, if the model is well predicted by its R-square value, correlation coefficient value. Okay. Here comes the question: what's the significance of this QSAR model? So far, we are discussing about QSAR, right? What might be the significance? How it is going to influence your laboratory experiments? Yes. Look at this question and say. So as I said before, in a drug discovery process, okay, it's tedious, right? And in vivo, in vitro procedure is tedious. How it is tedious? If I have an anti-cancer plant, I have to take the plant extract. Once I take the plant extract, what I have to do? I have to I take the plant extract. Either I have to treat the cancer cell lines or I have to treat the mouse and measure the values of the parameters, changing parameters. But what happens if I use in silico methods? I can use alternative in silico procedure, right? Like from the anti-cancer plant, rather taking the plant extract. If I know the plant's name, I can download the 2D structures of the compounds. The same way, rather knowing the influence of the plant compounds in mice, if I know what animals I'm treating or what cells I'm focused upon, I can take the 3D structures of the target receptors. So using the compounds for the particular receptors, I can train a model and predict the activity. So this is the significance. So QSAR models have been developed as an ideal alternative for biological experiments. So in reducing animal testing and designing clean and chemicals. So that's what I'm repeatedly saying, like from the collections of literature data. So this information you have to know. So my dear friends, I'm saying a word called data and information, right? So most of the audience might know the difference between data and information. Can anyone clarify that? What is data? What is information? What do you mean by data and what do you mean by information? And what is called knowledge? To make it simple, if I have
Data,
yes, please.
data refers to raw fact, figures or symbols,
Okay,
or the values which we collect.
Okay. So then what is information?
Information is the any result of any process, organization, or interpretation of the data.
Correct. So interpretation of the data gives us information. As one of our candidates have said, it is, let us make this very simple. Like 5, 10, 15. I'm writing these words, these numbers, 5, 10, 15, 20. So what is it? It's just a mere data. So what information you guys can collect from this data? Yes,
two odd and two even.
Yeah.
Odd and even information.
Yeah, correct. Odd and even. Or I can say even numbers are ending with zeros. Odd numbers are ending with 5. Multiples of 5. So I am deriving n number of information from the data, a given data. And accumulation of data, information will lead to knowledge, a pattern, right? So that information will try to make better predictions. So that's what's happening here. Whatever the data available in the resources, we scientists no need to perform the experiment again. So from the experiments already performed, we can derive the data and plan our predictions. So that's the beauty of machine learning today. Okay. And whatever the algorithms applicable, see, initially this information technology is helping for several sectors like banking sectors, administration sectors, but slowly after the advent of bioinformatics and cheminformatics, it tried to help in drug discovery process. So for several decades, it's helping. And with the advent of machine learning, the search is very fast now. Right? So you guys have to involve in QSAR techniques like machine learning techniques to equip yourself in the market, drug market now, right? Academic sectors now. So again, QSAR is not only restricted for biological applications. QSAR are applied in many disciplines. I will show the potential of QSAR in several job applications in a couple of slides. So QSAR is mainly applied in fields like risk assessment, toxicity prediction, regulatory decisions, okay, in addition to drug discovery and normal lead optimization techniques. So we have a huge significance of QSAR modeling. Fine. So now let us see about the opportunities in cheminformatics, means QSAR in cheminformatics. So if you guys are aware of QSAR and you have a huge role to play in as a cheminformatics scientist, like managing chemical databases and developing softwares. Okay, so what kind of industries you guys can place over is research and software development. And again, as a consultant in cheminformatics. So expertise in QSAR modeling can help them in consulting firms and research institutions. And you can play a role in the case of chemical engineer. So with the QSAR knowledge, we can optimize the process and formulation. So whatever the drug discovery process, or you have any number of leads, you can optimize those structures like chemical manufacturing and engineering. And the way computational chemist can collaborate with biologists in the case of drug discovery process, that is play a huge role in pharmaceuticals. The same way, guys can play a huge role in toxicology. Okay, as I said, QSAR is important for assessing the toxic drugs, okay, or toxic elements in the environment. They can assess the chemical toxicity, and they can play a role in regulatory roles. So that whatever the environmental approval, if you want to start an industry, they have to perform an EPA. Okay, according to Environmental Protection Act, whether the rules are managed or not. So they have to inform the regulatory agencies. So it's a huge field over toxicology. And again, environmental risk assessor, like evaluating the environmental impact, which influence the policy. So those environmental organization needs a huge impact of or huge involvement of QSAR. Then in the field of bioinformatics, so it's very visible, right? In bioinformatics, QSAR is important because in biology, we have huge amount of data relied upon the structure. Okay, structure of proteins and structure of drug molecules, and a number of drug molecules are there. So integrating the QSAR knowledge with the biological data, we can enhance the biomolecular research. So like biotechnology industries and bioinformatics industries has a huge demand of QSAR now. And again, as a biotechnological researcher, so if you even your core biotechnological researchers, QSAR will help you in drug discovery process. As I shown, you can bypass the in vitro and in vivo study, or it will help you to choose a particular compound in biotechnology and pharmaceuticals. And in the case of agriculture and food science, again, a food scientist or food chemist need QSAR in food safety and assessment. And an agrochemical scientist may need QSAR for understanding the agrochemicals, like impact of the chemicals on agriculture, pesticides, fertilizers control, and everything. And the quality control analyst. The same way, again, QSAR knowledge will help us to also evaluate the chemical patent applications. We can ensure the novelty. And as academic researcher professor, and the QSAR will help you to teach your students about the importance of it. And of course, the regulatory affairs specialist will help to know and have the knowledge of QSAR in making the regulatory affairs. So my dear friends, thanks for your patient listening. I'm happy to welcome any questions. Thank you.
Hello, sir.
Yeah.
Sir, I'm mainly interested to know what are the common or most commonly used bioinformatic tools for QSAR.
Okay. So QSAR, if you want to know, first of all, as I said, we have to know about the descriptors. Okay, designing the descriptors. For designing the descriptors, there is a thing called paddle. And for doing QSAR models, there are a number of softwares. Like you have a Python code or a dedicated software like QSAR and Veka and so on. So we will demonstrate that in the forthcoming, I think September session, like September mid of, we are having a Bioinformatics workshop, right? So in that workshop, we will perform over that. Am I answered the question?
Yes, sir. So actually, I just want to clarify one thing only, that
Okay.
if I have a selected group of ligands or drugs, and I'm checking their QSAR parameters, so
Okay.
it means I'm actually checking the ADMETs, or it is something else as well.
Okay. Apart from ADMET, if you look at the lecture what I provided you, I might have shown you descriptors. Just a second. Yeah. Fine. So admit is one among the properties, like log P, log T is one among the properties. Whereas you can even consider the H-bond acceptors or rotatable bonds, polar surface area. So any structure data can be converted into numbers. Okay, as a quantitative measure, you can use them as a descriptor to train your model. Fine. It's not only admit properties. Admit properties are majorly focused upon pharmacokinetics, correct? But apart from that, we have several other properties. And the current trend is about knowing about QM parameters, like quantum mechanical parameters like HOMO, LUMO. Hope you might have aware of that. If not, you may go and search over it, like higher occupied molecular orbital, lower unoccupied molecular orbital, HOMO, LUMO. So these are the parameters are used widely. Yes, Javad, do you have any other questions?
Yeah, thank you.
Yes, Dr. Anil,
Hello.
we couldn't hear you. Yeah.
Yes, sir, I have one question.
Yes, sir.
Suppose, sir, if I'm consuming 500 microgram of drug, okay?
Okay.
So what is the amount that the drug will finally reach the target site, sir? I have a small doubt there.
Again, it all based upon what kind of drug you're consuming, right? Like if you have oral drugs, you have to bypass about all the salivary amylases, and you have to bypass the acidity in the stomach. Still, until it reaches the intestine, after reaching the intestine only, it is going to enter into your blood capillaries. So until it enters the blood capillaries, you want to have an optimum amount to reach over there. Or if it is an intravenous drug, then again the drug dosage might be less. So the kinetics.
So where the rest of the amount of the drug will be lost, sir? The quantity of the drug.
Because in the intravenous, we are directly involving into the site of action, right? Sorry, not more than site of action. I'm saying about we are just reaching the blood capillaries. Okay. And the optimum dose or the very less dose will be at the site of action. We might be injecting directly into the site. So it all based upon the location of drug, where you are going to apply. Most of the successful drugs are ointments. So because they are topological. Fine.
Okay,
so
thank you.
Do you have any?
Dr. Chandrasekhar, you're muted. Can you please unmute yourself?
How about now? Can you hear me? Sorry for that.
Yeah. Please go ahead, please.
Yes, Dr. Anil, Hope I might have answered you. Any other questions? Or any clarification you guys need? We'll try to demonstrate this in the forthcoming seminar series. Yes, Pandya, Siddharth Pandya.
Yeah, so before going ahead, thank you, Dr. Chandrasekhar. I loved your presentation. It was really enlightening, and I myself have learned a lot. Your examples, your analogies, they were really amazing. It made many concepts easily understandable. And thank you so much for it. Thanks a lot.
Thank you.
Your presentation also slides were also very amazing. Like one can just have a look and try to know what you're trying to explain. So things didn't seem to be as complex as I feel it is. So it has been a very interactive session. Thank you so much. And your valuable insights are sure to have resonated with our audience, definitely. So yeah, I know there are questions lingering in the minds of the attendees. So we were, yes, about to start the Q&A session, but some of them came ahead and asked a few of the questions. Still, we have some and we'll address that. So while going through your presentation, Dr. Chandrasekhar, if you could look into slide number 23 and 24, where you have explained about the importance or the significance of QSAR analysis.
Just a second.
Yeah.
23 and 24. Yeah. This one, right?
Yeah. Yes, this one. We are saying that QSAR can be ideal substitution of biological experiments. So here I had this one doubt, since I am more into biological experiments and that's what I do in my company. Most of the time when we replicate a particular experiments, we have lots of data and they are not always the same. Like suppose I'm getting if I'm testing a drug, I'm getting, okay, 50 is the correct one. Next time when I do it in duplicates, triplicates, or conduct it multiple times, I'll get some other data points. Okay. So how much? Sorry?
Yeah, I got your question. The thing is, even you do triplicates and duplicates, the reliability of data. And we won't fetch the data in the particular order, right? We will fetch in range.
Yes.
So we will have an optimum range. So if our data lies over the range, we can rely upon the data. Fine. And more number of data we train. And again, we no need to depend upon only a particular data set. So here comes the quantitative measure, a combined action. Because none of the activity is based upon a individual. It is no more Bentham hypothesis, right? One gene, one enzyme hypothesis is no more. So we have systemic approach. Even it is a fever, it is because of several genes involved over there.
True.
So this omics approach, this QSAR approach will help us to predict a particular thing. And we guys are focused towards personalized medicine in near future.
Yeah.
As Human Genome Project, right is ahead, right? Human Genome Project READ is over by 13 years back in 1990 to 2003. Now again, Human Genome Project RIGHT is again going to over, and we are in the era of artificial intelligence. So huge number of data in hand will help you to make a proper predictions.
Yes.
So we can easily bypass animals and bypass all other smaller experiments. So again, we can't reduce the value of it. They are very, very important for collecting the data. And it can be very systematic if we have a knowledge of computers now. Algorithms now.
Okay. Okay. Thank you so much. Thank you. Thank you very much. Okay. Another question that we have is, so you might have, through experience, you might have known that there can be a lot of misconceptions and people might take the concept in a wrong way, like they might go wrong in few areas. So out of your experience of this QSAR and while teaching it and while working on it, what do you think, where do people most of the time go wrong? As in, what are the misconceptions and the myths most of them have in their minds.
Okay. So people are just jumble over data. By looking at the data, they got astonished and they got stuck over there. So no need to get panic over the number of data set we have. We just have to know the proper algorithms to manage over it. Okay. And many of us in QSAR make mistakes in choosing the descriptors. So descriptors have to be based upon the literature survey, what literature data we are just performing it. And again, we have to statistically validate the QSAR model. So any QSAR model you build have to need a proper validation. Okay. And a number of validation methods are there. Like as I said, R-square, and again, there's squared fit. And again, we have PCA. So these validation techniques have to be focused much. And the validity of QSAR is based upon how many test set you involve in. So, and again, increase more number of data to have a better predictability. So, Ms. Yasha, I have answered your question, I guess.
Yeah, sure. Okay, so moving on to the next question. One we had as in what is the difference between SAR and QSAR? So before going ahead, please explain what SAR is. And
again,
then what is, what is the
again,
difference between them, and how can somebody, if they want to master it, then how can they go ahead?
Okay. So the SAR is a part of QSAR, like structure activity relationship. So for that, I no need for a huge data set. It's enough to have a single molecule. Like if I have 5 drugs in my hand and 1 protein in my hand, I can do a simple docking. And I can understand how a structure activity will influence the relationship. Okay. Whereas QSAR involves huge number of data sets. At least I need some hundreds of molecules or thousands of molecules to train the model. So here comes the concept of training set, test set, and validation set in the case of QSAR. In case of SAR, the data set is less. Yes.
Okay. And so now people are so much interested into drug discovery, and you have explained in your slide that how different job profiles can use QSAR. Yeah. So people want to know that, do you provide any program or any course, any training where one can get trained about the drug discovery and all of that?
I think next week we have a 1 week workshop in QSAR. Means all different discoveries of cheminformatics methods. People can use over that, I guess. Yeah, cheminformatics and machine learning and drug discovery for the beginners. So whoever the thing, they can get enrolled in it. So it will be helpful for them to know the current techniques from different expertise in the field are going to deliver this. So guys, please make use of this virtual workshop. I think Dr. Mahesh or Ms. Yasha can elaborate about this.
Yeah, sure, I will. So I think we have come towards the end of our Q&A session. Thank you so much, Dr. Chandrasekhar. It was really interactive. I'm really happy. And this whole presentation and this session has definitely encouraged me to attend some of your freelancing classes, even if it is for NEET and GATE.
My God, thank you. That's a great word.
Okay. So soon, it's very exciting to say that soon we are going to have a virtual workshop on cheminformatics and machine learning in drug discovery, and this is for beginners. It's a 1-week workshop, and you have to register it through our website, that is www.neighborman.com, and your deadline is 7th of September 2023. The charge for the workshop is given on the slide. You can also find it on our website, and yes, you will also get a discount code. So for discount code, you can visit our website, www.neighborman.com/contact. So please do register to get trained for drug discovery and make a use of this workshop as much as you can. So right now, we have now reached towards the conclusion of our webinar. And I'd like to extend our heartfelt gratitude to Dr. Chandrasekhar for dedicating his time to this event. His insightful remarks are sure to have shed light on the minds of aspiring students, scientists, and researchers. We trust that our audience has gained a substantial amount of knowledge and clarity regarding QSAR. So dear audience, you will soon receive a feedback link from our Instagram team. Please give the correct email and name while filling the feedback. Lastly, please share the certificate on your social media platforms. Tag us and inform prospective candidates about our events. So before signing off, I'd like to once again thank each and every one of you and extend my best wishes for a wonderful week ahead. Hope to see you in our next webinar. Thank you so much. Bye-bye.
Thank you. Bye.